Greetings and welcome to the 2016 Coy W. Waller Distinguished Lecture. Thank you all for attending, and thank the Ford Center for providing such a very nice venue. I'm excited about hearing Dr. Mansoor Khan and appreciate his accepting our invitation for this event. Thank you, Mansoor, because he's a very busy man, as you will hear in a little while. However, first I'd like to have Dean David Allen from the School of Pharmacy provide some insight about Dr. Coy Waller, whom this occasion celebrates. Dean Allen?
Thank you very much, Dr. Rucka. Well, good morning, everyone. I appreciate your attendance today at our 12th annual Coy W. Waller Lecture. I'd like to begin by thanking the Department of Pharmaceutics and Drug Delivery for co-hosting the event. And I also want to thank Dr. Khan, who you will hear more about in just a few minutes, for serving as our 2016 Distinguished Lecturer. Dr. Khan and I worked together at Texas Tech, and in fact our offices were immediately adjacent to one another for a number of years. So I'm thrilled to see him here at Ole Miss. Since my first lecture as Dean in 2012, I've had the great honor of telling our audiences about the outstanding researcher that inspired this event. Dr. Coy W. Waller was an amazing man with a truly amazing legacy in natural products research. A North Carolina native, Dr. Waller was born in 1914. He received a bachelor's degree in pharmacy from the University of North Carolina, a master's in microanalytical chemistry from the University of Buffalo, and a doctorate in pharmaceutical chemistry from the University of Minnesota. Dr. Waller began his tenure with the University of Mississippi in 1968, serving as the Associate Director of the Research Institute for Pharmaceutical Sciences. This was also the year that our long-standing federally funded marijuana project began. Dr. Waller became the first full-time director of the Institute 2 years later. His focus on natural products as a source of new pharmaceuticals laid the foundation for the eventual creation of our National Center for Natural Products Research, the first of now 3 centers that are housed within the Research Institute for Pharmaceutical Sciences. Before retiring in 1979, Dr. Waller conducted research on poison ivy and the use of cannabinoids in glaucoma, and he helped edit bibliographies on marijuana and cocaine. In 1993, the university honored him by dedicating the Coy W. Waller Laboratory Complex, a natural products laboratory that housed our marijuana garden, medicinal plant garden, and greenhouses. Over the course of his career, Dr. Waller authored more than 50 patents, which were issued in more than 150 countries. He also received numerous awards. One of our former deans said that he was a beloved colleague who was greatly admired and internationally recognized for synthesizing folic acid. Dr. Waller was, without question, one of the most accomplished scientists of his time. We are grateful for his remarkable contributions to the pharmaceutical sciences. The school lost a great leader and research pioneer when Dr. Waller passed away in 1998. We are honored to recognize his extraordinary legacy with this event. I'm very happy today to see Ms. Deb Carnathan and Ms. Liz Thomas, 2 of Dr. Waller's 4 children, in the audience today, and I got a chance to visit with Deb and Liz right before we started. Deb and Liz, would you rise and let us honor you this morning? We appreciate your presence here today, and we are grateful to you and your family for your many contributions to the School of Pharmacy. I would like to now welcome Dr. Rucka back to the lectern. He will introduce Dr. Khan as our 2016 Waller Distinguished Lecturer. Mike?
It is my honor and privilege to introduce the 2016 Coy Waller Distinguished Lecturer, Dr. Mansoor Khan. Dr. Khan is a registered pharmacist, has been forever. Not as long as, well, I didn't mean that. And he earned his PhD degree in industrial pharmacy from the St. John's University School of Pharmacy in 1992. Dr. Khan subsequently earned his way to Professor of Pharmaceutics and Director of Graduate Program in the School of Pharmacy at Texas Tech University Health Science Center. In 2004, Dr. Khan joined FDA and served for over 11 years as the Director of Product Quality Research and a Senior Biomedical Research Scientist at CEDAR and FDA, where he led research teams on biotech products, small molecules, chemistry and stability, drug stability systems, and bioavailability bioequivalence. And as director of FDA, I think a lot of people don't know, but they are really a powerful position and make a lot of decisions. And Dr. Khan was very good at that. So we're really proud of him and honored to have him here just in that regard. Currently, he just accepted a new position as Professor and Vice Dean at Texas A&M Health Science Center, College of Pharmacy. Dr. Khan has published over 275 peer-reviewed manuscripts, 5 texts, including pharmaceutical and clinical calculations. I'm sure a lot of you will appreciate that. And quality for design for biopharmaceutical drug product development. Also 25 book chapters and more than 200 prestigious invited presentations worldwide. And those 200 presentations actually were over a period of about 10 years, so that's quite impressive. And also, he supported Ole Miss School of Pharmacy, and which is indicated by his accepting our invitation and his presence here today. Dr. Khan's research focus is primarily in the area of formulation, design, and development of biopharmaceutics. He's held several leadership positions at the American Association of Pharmaceutical Scientists, or AAPS, including chair of 2 sections within AAPS, Pharmaceutics and Drug Delivery, and the founding chair of Formulations, Design, and Development, FDD, now the largest section in AAPS. He's received the FDA CEDAR 2015 Outstanding A&D Reviews Award, over 10 FDA CEDAR Team Excellence Award, a number of different awards in excellence and exemplary performance. Dr. Khan received an Outstanding Alumni Award from St. John's University College of Pharmacy, as well as the Excellence Award from Texas A&M University Health Science Center. He also received the 2012 AAPS Research Achievement Award in Formulation Design and Development, and was elected both as an AAPS and an AAIPS Fellow. Today, Dr. Khan will be presenting a talk entitled Pharmaceutical Product Development After Approvals, Need for Clinical Observations and Corrections. Please join me in welcoming a recognized thought leader, Dr. Mansoor Khan.
Thank you very much, Dr. Resta. I would also like to thank Dr. Coy Walter and family for being here, and it's incredible what a difference the vision of an individual makes. And the Natural Product Center here is one of the best in the nation. I know the kind of work that gets done, and it's wonderful. And I hope there'll be more toy Walters here in the audience. Anytime I see students, I interact with them, I just feel so excited, and I just feel so humbled with all the opportunity that you have here. So I was here before in this campus a few times. Last time I talked about the need to develop products, because a lot of times we spend so much of time and energy and resources, but if you really think at the end of the day, what did the patient get, we really don't have much to show with our discoveries, because a lot of work gets done in a laboratory. They're excited, they get grants, they get R01s, NIH, and then publish papers, they get their promotion. But at the end, what did we get? What did the patient get? If you really see for all the work that we do, how much work we do. So I had challenged last time, I remember, that we need to interact with each other. There are silos there in different departments within the campus there, because some of the engineering folks, they do wonderful processing and the development work. Some of the chemists, they do excellent discovery work. We have natural product chemists, they extract drugs, beautiful drugs. They have shown a lot of promise there. We do all that, but eventually all these things need to be integrated with some nice parts and with key deliverables so that we get a product, and that product could be an IND, that product could be a new drug or a genetic drug or a biotech product. So I was talking more about how we can work together and discover. But today, I took this opportunity when I saw there would be a lot of white coats here in this laboratory. I wanted to take this opportunity and present some very profound aspects to you in your practice that you will see on everyday practice. You will be challenged a lot of times. We have been challenged a lot of times. A lot of times we don't have answers, but my desire is that you hear these things and bring solutions to those problems. There are problems, there are challenges. So initial aspect was how do you develop a product, but now you have a product. And the product, you are giving it to your patient. Your physician is telling you, this is my prescription, you're taking it, you're seeing it in a pharmacy, and you're seeing that the patient is taking a product, but you observe a few things, and I want you to be keen observers of those things. And I have numerous examples. Now I'm so blessed with all the opportunities that I've had to look at these issues, and I cherish the freedom that I have right now to come and talk to you and talk to everybody else, because a lot of times when you're in an organization, it's very difficult to talk. There are certain things you can't talk about, and you can't talk about that. There are restrictions. I just feel I'm liberated. I don't have those restrictions anymore because I'm in academia right now. So, but I will present to you certain aspects of those things just to open up your eyes and then look for those practical problems. So as I indicated, this is performance after approvals, right? So let's talk about it. So you have a product you are dispensing in your practice, right? And by the way, this is very, very important because we just have gone through our ACP accreditation now. We have the site visit there. I think you all know that the IPE, the interprofessional education, is very big, where the physicians, the nurses, the pharmacists, the other professionals, we all need to sit together and work on a problem. And that's what we have been doing on a daily basis in our FDA jobs. When we see some serious adverse events of certain products, that's where we were interacting with all these people, and so that we come to some solutions. So I will present some of those examples here. Right? So now, first thing, let's just review and the opportunities that we have here. We have people here, they are excellent chemists, be it a natural product chemist or a synthetic chemist. They are showing a lot of promise with their molecules. Oh, I extracted this drug. It has so much of medicinal value. Just now we were talking about, hey, some extract of marijuana or some plant might be useful for some condition there, right? They have discovered, but patients do not take this. Remember that. Patients do not take that chemical. What are they taking? They are taking this one. It's a biological licensing application, which is a monoclonal antibody, therapeutic protein, but eventually they're taking a dosage form, a tablet, injection, a capsule, syrup, suspensions, eye drops, ear drops, transdermal patch, that's what they're taking. And that's what this is about, the centerpiece here. So we get excited here, we get grants, we do everything here, but we don't know what to do next. We need to interact. We have beautiful chemists. We have a lot of people here, they understand this a lot. So when a drug is mixed with certain excipients, they can interact, they can make a lot of changes there, right? A lot of things can happen there. And then you have product properties. You have a transdermal product, you have an ointment, it has to have certain properties. I was in a workshop with one of your faculty members, Dr. Murthy there. They were trying to have some cyclosporin product there, the ocular product. So the contention was a brand company says a generic cannot be approved. A generic company says, I can make a product which is very similar to brand. Why don't you approve our product? So we are trying to look at product properties, a brand product and a generic product, whether they are equal or not, right? So, but we need to understand those product properties. Processing conditions, these are mixing, milling, granulation, compression, coating, lyophilization. Tell me who understands that better than an engineer? They do it all the time. Go to the laboratory, they do it all the time. I will tell you an incident there. Last year we approved, I was in FDA still, I was the lead reviewer, we approved a tablet which is 3D printed. It's like a printer you have, you're printing a paper, right? And paper was the drug, and the printing solution was the binder solution. So they put layers of drug and binder solution. You literally, quite literally, you printed a tablet out. And that product got approved. The very first time, I was excited about it because I was the lead reviewer there because FDA, they don't normally approve new technologies very easily because they're worried something else might happen. But I was excited. I came home, I told my family, I have 3 children, I told my family, we just approved a 3D printed tablet for the first time. My son listened to it. He didn't say anything to me. That was August 3rd. August 30th, or 31st, he joined Texas A&M in the engineering program. I was still in FDA, but he called me from Texas A&M. He said, Dad, that was August 30th or 31st. He said, Dad, you said 3D printing is new. I said, yeah. I said, come to our department. I will show you at least a dozen 3D printers right here with our faculty in engineering schools. Pharmacy school, we hadn't heard about it. But they are doing it day in and day out. If we had interacted with them, you can only imagine what we could have developed so far, right? So the processing condition, they understand it very good, but in all fairness and honesty, when I was an academic faculty, I barely interacted with them. I barely knew what they were doing. But now I'm seeing the need to interact with them. Stability. Normally the product, this is very profound, the stability work, and I challenge this pharmacy students too. Normally, a product is evaluated for stability at 25 degrees and 60% humidity in original container, which is sealed and it has a desiccant on it. So you open a container that has 500 tablets on it or 100 tablets on it. It has a desiccant in it, which is preventing the moisture uptake, right? And a cotton or a desiccant in it. But the way stability studies are done, you keep it in real time, 25-60, at 1 month's time point, take out some tablets and analyze it, see how much of the drug is there. Suppose it's a 100 milligram drug. After 3 months, you open a brand new sealed container again. You're not using the previous container, you're using a brand new open container again. And you're checking the content on it. Take some 10 to 30 tablets depending on the situation, right? And suppose out of 100 milligram now you have 98 milligram. Then 6 months later I do the same thing. Again, original new container there on it, my drug is 95%. 1 year later, it's 92%. So I keep doing until it's 90%. I mean, there are certain other requirements I'm trying to simplify. And then I determine an expiration date. I say the product expires in 2 years. And that's what my young friends are seeing in the pharmacy. Hey, its expiration date is February 2016 or March 2016, whatever, right? But how was the stability? study done on original container, sealed container, but what are you doing in pharmacy? What you're doing in pharmacy is, out of those 500 tablets, you dispense 30 tablets, but 470 are sitting in a pharmacy forever. Then the next patient comes, give me another 10, 20, 30 tablets. The medication is, but what happened, the first thing, I used to work as a pharmacist, so I know this thing very well. First thing I do, I take out the cotton, and I open the seal. I take out the cotton, I just throw it out. I take out the desiccant, I just throw it out. And that's how my container and the tablets are sitting there. Who has done the evaluation for that? Nobody has done the evaluation for that. So the end result is, in the real condition, patients are getting 10 to 30, 40, whatever tablets you are in a vial. We don't do stipulated studies on those things. So what I'm trying to say is, products can be different from what is promised on the label sometimes, right? So just pay attention to it. So container closure system, whether plastic or a glass, can make a difference. Scale up. You made a few tablets, got your approval, but after that, you made a big batch of product. And you made a product here, you made a product in India, you made a product in China, you made a product in all kinds of places there, right? But you have to scale up. So it can bring some variables. So don't see it very well. The analytical methods, so this is what I've seen a lot of times. The methods we use, a lot of times they are not validated. So if I'm doing an HPLC retention time of 30 minutes, sometimes it shifts to 15 minutes, 14 minutes, 12 minutes, right? It's not very, very consistent and constant, right? So we need to pay attention to all these things to have a product. So sometimes things can change here. You're trying to mix, something change in the mixer, something change in the excipient. You're trying to add an excipient and the supplier changed. So a lot of things can change, the product can change. I'm saying all this and I spent time on this slide because that's the reality you face. The product can change. So when you are giving a product to the patient, sometimes it doesn't work. You know, it's antihypertensive, you know, the blood pressure is not going down, or diabetic drug, it's not working, or some other medication is not working, or sometimes there are some serious adverse events. So think about product also. Don't just think about other factors. So something you need to think. So I will give you numerous examples. These are real practical life examples. So now a doctor from Cleveland Clinic had a liver, writes to us and says, I have been giving metoprolol for a long period of time, but this brand of metoprolol or this metoprolol just doesn't work. Now what happens when they write something like that? It goes to a MedWatch report, and from that report it goes to the very same reviewer who approved this product in the first place, and then we ask the reviewer, hey, did you miss anything in it? He said, no, I don't think I missed anything. Then why is that patient not benefiting from it? Hey, that patient is not benefiting because he's like me, an Indian American, he's vegetarian, and so his food habit is different, yes. Or he's an African American, or he's Hispanic. It's not working because of those genomic differences, right? That's what happens. A lot of times it happens. So, and then now we seek funding. Hey, let's understand this genome. They sequence the whole genome right now, right? But the patient, end of the day, it still gets, so when we look at those tablets here, instead of having this dissolution in the original tablet, they have this dissolution. The drug is not dissolving very well, doesn't get absorbed very well. So what they did, I traced back the papers, what I did, in the actual product, dissolution was going down, they changed the method. Hey, product is still the same. We didn't improve the product. So why was the product failing? So we wanted to go a little bit further and find out why the product was failing. So this is what we did. So here is the product. These are some small beads. You coat them, you make them sustained release, so the drug gets released very, very slowly. So you take these small beads, and you take all these beads together, and you compress them. And when you compress it, you add an excipient called a cushioning agent, and you compress it, so the membrane, thin film around, remains intact because you have a cushioning agent there. So you have a cushioning agent when you compress it. But if you don't add a cushioning agent, it gets ruptured, right? So when you see this product, this is what happens. So it's a very simple test. The pharmacist can do it. You put the tablet in water, the beads come out. When the beads come out, you see, in some of them, they are intact beads. So I can say that, hey, you know what, these are good beads, but look at here. They got compressed. When they got compressed, they don't release very well. So the problem was in the product. So immediately we corrected that problem. And then, so initial investigation said, oh, there's no problem with the product. But another group of people looked at it, yeah, there was problem with the product. So we put it out, right? So when you see a patient not benefiting, not getting affected, collaborate with your colleagues and try to find out, right? So spend some time on it. Don't just say, hey, it's a genomic difference, or the patient is big, or is overweight, or whatever, right? Problem could be here. I'll give you another example. There was a media report sometime in 2008. It was in most of the mainstream media. There was a question about brand and generic product. Hey, this product is not the same because this is the active product, and generic is allowed to have different shape, different size, different excipient they're allowed to have, right? So a generic product. So what they were saying, there were a lot of complaints. In the media, they were saying a lot of complaints. We took it, this side, again, it goes to the same review, oh, there is no problem there. But once what happened, a lady, her husband shot himself with a gun, called from Houston, and she sent a note to commissioner, she sent a note to me, to commissioner, with a copy to Senator K. Bailey Hutchinson, a lot of folks there around there. This is what happened. But she believed that her husband was taking a certain kind of a product and then he changed the product. She believed that that was happening with the product. But you know, it's a human aspect of it. Somebody wanted to look at it, so we looked at it, right? So, products can be designed in different ways. One of them could be coded so you can release a drug slowly, or another one could be in a matrix, right? But they look at certain properties. Does it have the same content? Does it have the same release profile? That's how the product get approved. So here, when we looked at it independently, not to the same reviewers, independently, this is what we saw. The drug was releasing differently. Immediately that gave an idea that since it's releasing so differently, maybe they're getting a different response. If you get too much of drug here, it's a 24-hour medication. If you get too much drug in the beginning, later on you may not be getting drug in, right? In 24 hours, the first 2 hours, most of the drug came out, got absorbed. What happened the rest of the time, right? So there could be a difference. But they said, hey, laboratory investigation doesn't necessarily translate to human data. So we said, all right, let's look at human data. We did this study, actually human volunteers. Took 24 subjects, gave both these two different products, right? Took out the plasma concentration, analyzed it. You learned that, right? You learned that in pharmacokinetics, right? The students learned it in pharmacokinetics, right? How do you do bioavailability, bioequivalence test, right? You administer the drug, extract some plasma and analyze the concentration, right? Plasma concentration versus time. So we did that. I'm sure you can do it in pharmacy school here too. We did that, right? So that's what we found. Now at first, if you just see it, in the time 1 hour itself, there's a lot of drug coming out here. Versus this is the original product. Look at the drug coming out and look at the drug amount coming out. So it's really going well with the in vitro data of dissolution that you had, right? Too much drug in the beginning, here they have too much drug in humans too. So we wanted to see whether these two products are equal or not. Now, a confidence interval should be 80 to 125%. I think you all know, right? For bioequivalence, the Cmax should be 80 to 125%. And area under the curve should be 80 to 125%. What that means in a nutshell is, if it is between 80 to 125, these two products are equal. If it is not between, so you see it bombed. So what we did, we called the company. Hey, this is what we saw in your product. Immediately the product was pulled out of the market. Immediately was pulled out. But if somebody was alert enough to see these things, they did the study, that's how the product. If we had not seen it, if the pharmacist had not paid attention, if it had not gone to media, we won't probably have paid attention. A lot of patients might have been taking this drug, right? So what we did, we wanted people to know this. It was the very first study of its kind that we did in FDA. So So we wanted people to know just about everything we did. We wanted to be completely transparent with the people. So we published in New England Journal of Medicine. We just said, hey, you need to be careful, and got the product out, right? So again, it's the end result of somebody paying attention when the patient was taking it. Now, tacrolimus, the radio talk show host. Radio talk show said somebody took a liver transplant and organ got rejected, and then there started a lot of discussions about it, right? We wanted to look at that product. Why are they complaining so much? Look at that product. So, if the compound is very crystalline here, the bioavailability is very, very low. This is the plasma concentration. Bioavailability is very low. But if you, same chemical, you process it differently. You make it amorphous. Bioavailability is very high. But you know that amorphous compounds are thermodynamically not very stable. If you keep it aside, it tends to go back to the crystalline form. Right? So they're not very stable. So dissolution was going down here because the amorphous was getting converted back to the original form. So what they did, they added some surfactant in it, again changed the method to bring it up, but that didn't make the product better. It just changed the method, but the product is still the same. We developed a method, we identified what the issue is, and we removed that product from the market. We removed that product. Now this is very dear to me. I use this example a lot. These are the products that you actually see in your pharmacies. Many of us have children. A child has cough, cold, allergy. What do? We go to Walgreens, CVS, some other place. Hey, this is for cough, this is for cold, this is for allergy, right? These are the products. These are the products. And these products, some of them, they're good for 24 hours, so they write out allergy treatment for 24 hours, cough treatment for 24 hours. What are we doing, my friends? Chlorpheniramine maleate was approved, but chlorpheniramine tannate, FDA did not approve chlorpheniramine tannate. But tannate, they are long-lasting, 24 hours. But what are we doing? When just a child, 2-year-old, has a cough or allergy, what did I do? I brought that and I gave this medication and I said, my cough is relieved, my cough is treated. And I get a 24-hour, are we treating the cough? Who says we are treating the cough with chlorpheniramine tannate? Where is the data? You are just putting that child to sleep. It's an antihistamine, right? You sleep, right? What a relief. They struggled for 30 years to challenge them to take this product out or give us the data, but they fought back. They said, hey, the pre-1962 drugs were our grandfather, and by law we are protected, their grandfather. But what they did not know, was the law. And I mean, that's where if you have people who know or qualified, who are passionate about it, I mean, I took it personally. I have children. I have 3 children, right? So, if it is time release, it's not grandfather. They're very clever. They don't write time release product on it. If you see those products, nowhere it's written time release. It's some name. It's not time release. But we are scientists, we can understand, pharmacists can understand. If you see the ingredients there, the kind of ingredients you have are very similar to an extended release ingredient. Right? So all we had to do was, even though it's not labeled as extended release, the product is extended release. So all we had to do was do some simple test. You have dissolution equipment right here. Sustained release, you have dissolution like immediate release, you go like this, right? And even after 24 hours, you still have some tablets remaining here. That's it. We made our case. So we sent the report. We sent the report in November of 2010, I think. By March of 2011, in 3 months, 500 products removed from the market. And you know what? You remove one product, you get challenged. The challenge at the end. take you to court. 500 products came out, nobody challenged FDA. It was based on science, based on data, based on your observations, right? An example. Products were out, and so I felt so not necessarily we were trying to take product out. All we were asking is, show the data to the reviewers who are eagerly waiting to look at your data, and they will approve it. Because it's very profound, right? So it's an example. One example here, Alendronate, Fosamax, a lot of mistake, right? Especially elderly, osteoporosis, a lot of mistake. Report in New England Journal, by the way, New England Journal of Medicine, for those who don't know, is the highest impact factor journal. All the journals, more than Science, more than Nature, more than anything else. Highest impact factor. You see an article like that, you have to pay attention. Pay attention, right? They are saying it's causing esophagitis, it's burning here. So you have a patient that's coming to you, is taking this medication, and says, you know, it burns here. It burns here. Don't tell him, hey, you are Indian American, African American, that's why it's burning to you. Right? You have all this genomic difference there. Pay attention. Why is he saying that? Is it one person saying, or 2, 3, 5, 10, 20 people saying that? 10, 20 people are saying, you need to pay attention to it. So when we saw this, when we saw the complaints, when we started looking at it, you see the esophagitis in esophagus. You have the GI transit time around this region of 15 to 20 seconds. The product is designed not to disintegrate very quickly. That's a good product. So they want an intact tablet to go from here to here. Because once it disintegrates the surface area and it sticks like a leaf. The properties of the compound is such, it sticks. Very acidic compound, it sticks there and it causes problems there, right? So it should not disintegrate fast. But sometimes our common understanding is if something disintegrates faster, it's acting quicker, it's acting better. For an osteoporotic patient, you don't need that. It's not a migraine. There's a difference. In a migraine, yeah, you need something very quick, right? So here in this particular case, you don't need that quick. So we look at, all right, what is the product doing? Look at all these products here. So this product, 1/22, it should not disintegrate. Very simple test. Should not disintegrate. Look at these products. And they thought their products are better by disintegrating faster. So what if you find out, hey, this particular patient is complaining here because you saw the New England Journal article, you saw the tablet, you know pharmaceutics faculty like you in the school, hey, you know what? Maybe something is happening. Talk to the faculty. It's a 2 minutes test. By the time you prepare and all those things, in less than 1 day, you complete everything. Right? So, published, more details are published in this journal. When I was a director there, I was really blessed. You know, it's like a little kid likes candy, you put him in a candy store. I was a hungry scientist. A lot of information available to me. So one of that was, I wanted to do a recall, because there's some products that you have seen, they were recalled. So I wanted to go and look at all the recall reports. I said, I want to stop all activities. I want to see this. I haven't been able to publish this, but again, I'm able to at least talk. 7,200 recalls in about 6 years. So that is about 1,200 recalls per year. You know, each recall, you know what it entails? I guess you can only imagine. 35,000 pharmacies. The product is there. You're telling all those pharmacies, this lot of this product is not good. Don't sell it. Who is complying with it? Who's going? Who's investigating? Who's checking? And I bet you a product might be sitting in my own home or in my own school or in my own family. They don't even know what recall means. Somebody is using that product. It's not easy to take out. So recall is not a good thing. It's not a good thing for the sponsor, for the company, not a good thing for FDA, not a good thing for the patient. It's not good for anybody. The products are getting recalled. So I wanted to see why these recalls are. Look at that. Impurities and degradation products, 150 recalls. Failed dissolution. When these recalls happen, some patient is taking and complaining and coming and telling you, to pharmacists in white coats, I'm having this problem, my diabetes is not getting better, my hypertension is not getting better, this is not getting better. You need to pay attention to it. Right? So I wanted to look at, they were sub-potent products. There were 223 recalls from the period that I saw, right? Now, sub-potency, think about it. I mentioned, remember I told you about stability studies, how they're done, right? In the real life scenario, a lot of things have changed. So when they evaluated a product, right, was sub-potent, okay? So a lot of things can change in a product. I have described those things here, but a lot of things can change. For example, an excipient can change, and I'll show you some examples. They just change an excipient, but it changed the stability of a product. They change a process. It changed the stability of a product. So there are some examples that I will show you in the coming slide. There were 70 recalls for levothyroxine. So I asked my clinical pharmacy colleagues here, really what I want to do is I want to go on rounds with them. How the patient is getting treated. Look at the charts of the patient. I'm giving ideas so that the basic science and clinical faculty can work together. Go sometime with clinical pharmacy faculty. Look at the chart. See how the dose is getting adjusted. Hey, levothyroxine, you're giving 100 micrograms. Suddenly you change it to 88.5 or 75 microgram or 50 microgram. Why did you do that? You didn't do it previously, you just did it now. So maybe there's a problem with the lot, right? If you're seeing changes, you need to pay attention to it, right? I would really like to see that. So we've seen a lot for levothyroxine. Look at that. We wanted to do a stability study on that one. Look at that. This product, it should have 90%. Look at this product, initial. Immediately we removed that product. But the problem with levothyroxine is, if the potency is 90%, a newer 100 microgram drug had more strength than an older 112.5 microgram drug. So physicians were having difficulty adjusting the dose. So what we did, this is a unique example, where we changed the potency requirement to 95% instead of 90%. So this particular product, because we had approved, it's not easy to take the product out, so we changed the requirement to 95% potency requirement. That's how we got the product out. Right? Now, so we did some investigations. Actually, if you write down my name in PubMed or whatever journal, you will see these articles there. So why was levothyroxine, some product it was degraded, some other product it was not degraded? We wanted to investigate that, right? So it is a method we developed. So this is levothyroxine, it has 4 iodines in it. If you knock off 1 iodine in it, you have a peak. This is T3. Knock off 2 iodines in it, it's T2. So I think Dean Allen would know, had a student, Rocky Shah, and Rocky was a Texas Tech student, but she was working with me in FDA too. So she developed that method actually. So by one single method, you can see the original levothyroxine and all the breakdown products. So we had to have a stability indicating method. So we developed the method first, and then what we did, we looked at certain excipients, and these excipients were used commercially in the product. It gets hydrolyzed. If there's a moisture there in it, in a product, levothyroxine breaks down. So you see these excipients here, they were picking up, this is the weight versus humidity. They were picking up a lot of moisture. So they selected excipient that picks up moisture and made a product. So when a container gets opened, this excipient is picking moisture and it's degrading levothyroxine. So you should avoid those excipients. But these sponsors didn't know. Actually, the product had been on the market for a long period of time. So if you see these excipients here, see they're degrading the product very fast. So we made a post-marketing correction of it. Post-marketing correction, and so we changed those things, right? This is gabapentin. A sponsor called and they said, we have $200 million product out in the pharmacies today, And please allow us to extend the impurity level. It was 0.4. This derives for degradation product, 0.4. Please allow us to change it to 0.6. It's an impurity, degradation product. So we wanted, and this causes cancer. This is one of those few products the impurity causes cancer, right? So we wanted to see why this is happening. We told them, no, you can't do it. So essentially you can see, depending on the excipient they select, some of them that they were degrading very, very fast. That impurity was forming a lot, right? So again, they had to change the excipients there. Nimodipine. I'm giving you examples here, right? Nimodipine. There was a recall. There's a liquid-filled capsule, but there were some crystals in it. Where are the crystals coming from? Then there was suggestion, oh, they didn't filter properly. Oh, they didn't make the product well. My friend, they made the product well, but again, like I mentioned to you, the stability, different temperature, humidity condition, is it not conceivable that truck was going to some cold region, it's chilly a little bit here, right? The product was made when it was 80 degrees, 70 degrees, whatever. The temperature has lowered, because of that the crystals come out. And sure enough, we prepared, we created some formulations internally, right? And exposed them to different conditions. And some formulations, they were showing a lot of crystals here. So immediately we knew what kind of formulation will show crystals when temperature fluctuates. And there are other products, they don't show crystallinity when the temperatures fluctuate. A good sponsor would get this one. A sponsor who doesn't understand would get this product. We recall that product. So essentially, it was changing from one form to another form. That's how the product was crystallizing out. Now, this is a very interesting story. When people complain, we look at the product and say the product is bad, we take it out. Here is a product from Harvard, Dr. Preston Mason from Harvard, from Brigham Young. Preston Mason called, he went to Congress, 50 congressional leaders. This is what he said. You know what he said? We are Wild West in FDA. FDA has gone crazy. We are Wild West. The product has 20 to 30% methylated impurities, and FDA approved it. And generic product, specifically product coming from India, right, has 20 to 30% impurities. Now my boss gets called for a congressional testimony on that. I was very shocked. So we wanted to see how could FDA approve 20 to 30. Allowed levels are only 0.1%, 0.15%, 0.1%. 0.1% is allowed level. How can a company from India or any place have 20 to 30% impurity? Actually, impurity costs more money than the drug to make it. How can that happen? But he went to Congress. So we wanted to see it. First thing, what he said was, these companies, generic companies, is the tertiary butyl groups in this method here. They said they were in a rush. It's a cheap company. It's a cheap product. They did not clean it up very well. By some postulate, the O-butyl groups got converted to O-methyl group because the impurity was O-methyl. I mean, you all take chemistry. How can you get O-methyl from O-butyl groups? Fundamentally, the concept was. Then I looked at his background. He's a molecular biologist. Why is he looking at a product? Right? So I said, but the next question, so I knew that the postulate was wrong. I knew that. But the next question was, how did he get 20 to 30% impurity? I think somebody from this audience will tell me now. It's an ester impurity. Atorvastatin is an acid. What do you need to do to an acid to form an ester? Solvent, alcohol, very good. So I looked at his method. He prepared stock solution in alcohol. He put the acid in alcohol. The molecular biologist who goes to Congress, right? So I did. We went to the laboratory. In that acid, we put atorvastatin in it, and sure enough, with 4 hours, 5 hours, 6 hours, you see this Atorvastatin, and this is the impurity he found, and he said, hey, he has gone Wild West. So what we did, he used the wrong method, used the wrong solvent, used the wrong starter. We did with the right method that was approved. We looked at all the products. See what we got. This is the approved 0.1%. You don't have 20, 30%. None of the products. This product had a little bit more, but still it's within the limits. So sometimes we get comments like that. They're not right. They're not right. I don't know what the motive was, they were not right. But again, by the way, so what we did with this, so I showed you the news that was February 26, 2014, when President Mason went to Congress, and Bloomberg's news covered that, right? And everybody was, I was up almost all night. I was up all night, trying to figure out what happened, right? That was February 26, 2014. Look at this. March 25th, 2014, 1 month. Actually, 1 day less. The same Bloomberg's news writes down, there's no problem. We challenged Harvard. There's no problem. And what we did, again, we wanted to be transparent. We published. We want people to know. Now these my friends here from South America, right now, the generic products of vancomycin, they're not effective. Everybody gets worked up. These products are not working. Complaints. We look at the data. These people from South America, product expired 3-4 years after the product expired. They want to do a study on that, and then they say it doesn't work. Why would it work? The product expired a long time ago. And that product is not even in the US. Some other company made it. But they landed on FDA. That is our fault, our problem, right? So immediately we did, again, we had to spend time and resources and all those things, because if media says something, we can't be quite responsible, right? So we look at it, did the study. Again, they said, just like the Atorvastatin folks that led in impurity, they said, oh, there is a lot of CD3 impurity. Hey, these were all the approved products, none of them had that impurity. Again, you have to develop that. So what I'm trying to say is sometimes when the patient complains, pay attention to it, you might be able to figure out. A lot of times we're able to figure out, but there are times when there are other motives. I know one company, they just changed the capsule of the body and the shell, right? The top part and the bottom part. All they did was change the color of the bottom part. It was Pfizer or some big company. All they did was change the color of the bottom. Man, you got so many complaints, the product is not working. They didn't do anything to the formulation. So sometimes you hear things like that, right? But sometimes the reasons are genuine. So I want you to be alert as pharmacy student. Product could be different. There are so many variables there. You saw that, right? Temperature, excipient, process, where it is made, analytical method. A lot of things can change. You need to pay attention to it. So I would think my brother called me. My brother is a cardiologist in Ohio. Sometime ago, just 3 months ago, 4 months ago, he was given heparin to the patient. And he said, Mansoor, I don't know why I have to increase the dose a little bit for it to work. And he kept experimenting. He figured out after 6 months, if we double the dose, it's working the way it was normally working before. But he had to double the dose. So I don't know. I came out of FDA by then. But is it not conceivable that somebody by mistake has added the side drug? That's why you have to double the dose for that particular month, right? So if you see things like that, pay attention and think about it, or interact with your colleagues in the pharmaceutics and in the pharmacy school. Try to have that attachment even after you graduate. Try to have the attachment so that you can figure out what happened there. Because eventually what you want is the optimal response for the patients. Right? So I would like to conclude. I think I have given some examples, but here is what I want to make. It is critical. The product and process needs to be understood. sorry about that. This is optimal therapeutic response. Methylated impurity, not the one. This is not of any concern to us. I had changed the slide, but I forgot to add it here. What I wanted to say was, when you see an adverse, I had a bullet there, but it's in my other slide. I forgot to give it to Dr. Recker. What it is when you see adverse events on a number of patients, then look at product. Look at product as one of the contributing factors. Don't just think that, hey, this patient is this different, that patient is different. A lot of times, industry is very good at that. And FDA is also very good at that. You deflect the problem. The problem is here in the product right here. But you're deflecting, oh, it is happening maybe because he's on this vegetarian diet or whatever. His genome is different. Like I said before, we explore the whole genome there. But the problem like this that are sitting here, those problems will not go away. No matter how much research we do, those problems will be there, right? So I want you all to pay attention to it, and I think it's a tremendous opportunity. That's what the interprofessional education, when a doctor finds, when a clinical pharmacist on their round finds a problem there, interact with the basic scientists and figure out, the chances are it's there in the formulation somewhere. With this, I will stop, and I would really like to thank you all very much for giving me this opportunity to be here with you. Thank you.
We have time for just a few or a couple of questions. If anybody has any questions, there's some microphones. If you have a question, microphone on each side of the rows there.
Thank you very much for excellent lecture. And it's very important what you said. Everybody blames on the genetic variability. Based on your experience, these are the approved drugs which go through the process and manufacturing. Any comment on the pharmacists are dealing with a lot of products which are called dietary supplements. Some people debate about regulation and not regulation. Based on being an FDA in the School of Pharmacy, what is your take on it? There is no dissolution, there is no proprietary thing, people are mixing different formulations. So can you give us your opinion about it?
I would be very happy to do that. Actually, you all have an opportunity to be champions of that because of the center that you have here. I'll tell you what happened. With my own experience, I was in Louisiana before going to Texas Tech. So we looked at coenzyme Q10 that was commercially available. So we looked at all the products that were commercially available. It was a 30 milligram thing. None of them had 30 milligrams on it. The maximum we had was 29.5 by the method that we had, but some of them had 6 milligrams on it. But on the label it was 30 milligrams, right? And then one of the companies, I happened to call them, and I went to the company, I saw they did add 30 milligrams. They showed me all the calculations for the million batch they were making, they did add it. But then I asked them, did you look at how the product degrades? They haven't done. So if they add a product, and for the photo degradation, you expose it to light, it degrades, right? That was one issue. The second issue that my own experience that I'm telling you. The second experience, I think this is for your knowledge. About 2 years ago, we decided in FDA, 2-3 years ago, we decided in FDA that, all right, we will get some of these products that are there in the market. Let's look at them. So there were 13 products we got to evaluate. They were for erectile dysfunction, they were for diabetes and hypertension, right? All 13 products. For the one that said it's a natural product, they said it's good for erectile dysfunction, all 10 out of 10, all of them had Viagra mixed in. Okay? And then the diabetic drug, there's anti-diabetic there in it. So there are a lot of players out there who really want to make money. So the genuine scientists, well-meaning scientists should step up and shout by examples like that. I couldn't publish those things. A friend of mine, I think I know some of you know that, what's his name, Bill Gurley from Arkansas. He published that, but I've heard a lot of challenges. I mean, I couldn't face all that crowd and the media attention and all that. But I want some responsible people to take this issue. There are a lot of products they want. I have another study that came to Johns Hopkins for a clinical study. They wanted them to look at a clinical study, but luckily those scientists were very good. Before they did the clinical study, they evaluated the product because they were trained for anti-cancer activity, natural product. They laced it with a cancer compound there in it, and they wanted them to do a clinical study. What result will you get? And then they were hoping that, hey, this natural product has this efficacy, and this study is coming from Johns Hopkins. But luckily they were smart, they were alert, and now, so because of issues like this, I think these products need to have oversight, these products need to have what is being added there in it, who's adding it, how do you analyze it, can you say for sure what they've added is what's there in it? We need to have assurance of that, we need to have those quality characteristics. I think you are in the best position to fight for it. And I'll be happy to join you in that fight. But what our patient, whatever we tell them on the label, they deserve it, they should get it.
Okay, thank you. Maybe one more quick question, otherwise we need to go ahead and conclude. Anyone else? Anyone? Okay. Well, I do want to express our appreciation to Dr. Mansoor Khan for taking his valuable time out to come and give us some insight on his experiences. And I think it's all relevant to what, for the professional students and for graduate students and for all of us. So anyway, thank you very much, Dr. Khan. We appreciate it, and we'd like to just present you with a small token of our appreciation. Okay, thank you. You can go ahead and